Hi guys, you're about to watch a 1 hour video on cell structure and function, and it's not even complete. There's still a video to go on chloroplasts and mitochondria. So I thought I'd take a little bit of time to justify the length of this thing that you're about to watch. And it all comes down to this. It's not long just for the sake of it. I really do believe that there's two ways that you can know your subject. One is the quick and easy way, where you know what you need to know as quickly and as efficiently as you can do it, and then you reproduce that at some later point in time. Or you could really get your hands dirty, understand why things are the way they are, and thereby know it more deeply and better, so that when it comes to applying that knowledge, you're in a better position to do that and therefore get higher grades. So if you do make it to the end of this, well done to you, and I hope that you find it useful. In the last episode, we discussed the evolution and the reason why we have the different organelles. In this episode, we are going to start looking in detail at the structure and function of specific organelles in the cell. We're going to begin with functionally what ties them together, i.e. giving them a purpose, some kind of related function to which they all contribute. And then we'll start looking at specifically their individual structure and how that relates to the function that they have within the larger overall purpose of their being in the cell. So as we discussed in the previous episode, is that the nucleus, the endoplasmic reticulum, the Golgi Apparatus, and lysosome and vesicles, they all have this collective purpose of lipid and protein synthesis. So that's what we're going to look at. Many cells, critical to their functioning is the production of specific proteins. So we've got 4 different proteins and different locations in the cell. So we have a cytosolic protein here. We have a protein that exists in the cell, but it functions in a particular organelle in the cell. Then we have a protein whose function is in the cell membrane. And finally, we have a protein that its function is outside of the cell. It's an extracellular protein. It could be an enzyme, it could be a structural protein, it could be a hormone, but it works outside of the cell, and so it needs to be secreted. So we have these 4 different proteins, and what we are going to do is look at the role of the nucleus, the endoplasmic reticulum, the Golgi apparatus. We're going to look at the function of these organelles within that scenario. We have these different structures in the cell. It's not a coincidence that they have different structures and different functions. So one of the things, and one of the reasons why we are going to focus on protein synthesis is because it's the proteins that exist within these organelles that give them their structure and that give them their function. And therefore, that gives them their different identity to us, okay? So different appearance, different overall thing that they do in the cell, all of those properties are bestowed upon them because of the protein content that they have. And the reason why we say that the nucleus controls all the activities of the cell is because all the proteins that give these different organelles their different functions, all these different proteins are encoded for the most part in the nucleus, in the genes that exist within the DNA that exists within the nucleus. That is why we say that the nucleus controls the activities of the cell. It doesn't physically actually control any of the activities of the cell, really. It's just because it has the genes that get converted in, or that get used to make proteins, and those proteins exist within specific locations in the cell, and it's the proteins that endow the different organelles with the different functions in order for them to contribute to the overall function of the cell, which, as we said in the last video, that the function of the cell contributes to the function of the tissue to the organ to the organ systems, the organ system to the organism, and so on. Okay, so let's then go back to the nucleus for the origin of all these proteins. Okay, so we begin in the nucleus, and all those red squiggly lines represent chromosomal DNA. And for the synthesis of a protein, remember that if this section of the DNA right here corresponds to a gene, then, and that gene corresponds to one of these proteins, okay? So it could be a gene for this protein or the gene for that protein. What will happen is proteins will start to congregate around that gene, around the promoter or the operator. I can't remember now which one it is that they physically bind to. But that gene, because of the collection of proteins, which is going to include RNA polymerase, transcription factors, helicase, and helicase will be responsible for unwinding the helix and unzipping the helix, so the two strands come apart, the bases will be exposed, okay? And where the bases are exposed, this is where RNA polymerase will then use one of the strands as a template to synthesize the RNA. Okay, so I'll use green for RNA. And so one of the strands will be used, complementary strand will be synthesized using RNA nucleotides within the, or complementary to that gene or strand. And then as the strand rewinds again, the RNA will come away, and that is what we know as mRNA. So the mRNA is now produced, it's not there anymore, the gene rewinds, rezips, okay, so rezips and then rewinds, and then we have it back again, and all the proteins go away and they go to another gene because it's time to transcribe that gene. So we have the mRNA. Now, the mRNA is within the nucleus, but it needs to bind to a ribosome out here, either in the cytoplasm. So obviously some ribosomes are cytoplasmic, not bound to the endoplasmic reticulum. So I'll just draw a few of them here. Okay, and the mRNA now exits the nucleoplasm, exits through the nuclear pore and enters the cytoplasm. Okay, so mRNA is now going to snake through the nuclear pore. Again, we will come back to the specific structures of all these different parts that I'm mentioning, but before we look at that, it's very important to appreciate the bigger picture into which these organelles all fit, okay? And we've got ample time in which to discuss all of this stuff. So the mRNA snakes out of the nucleus through the nuclear pore, and it's now moving through the cytoplasm. And what will happen is that if it has a particular sequence in its bases, if it has a particular sequence that directs it to the rough endoplasmic reticulum, it will bind to a ribosome at the rough endoplasmic reticulum. If that sequence of bases in the mRNA is missing, then that mRNA will continue to snake around the cytoplasm until it binds a cytoplasmic ribosome. Okay, so let's look at those two scenarios and then what will happen. So if it lacks that sequence that directs it to the rough endoplasmic reticulum, it will bind to this ribosome right here. Okay, but if it does have that sequence, it will bind to a ribosome on the rough endoplasmic reticulum on the outside. Okay, so yeah, now if it's a cytoplasmic protein like this, so let's say this is an enzyme, perhaps it's an enzyme that's involved in glycolysis, glycolysis being a metabolic pathway that happens in the cytoplasm of cells, so that could very well be one of the enzymes involved in that process, glucokinase, for example. Okay, so if it's that, then it needs to be synthesized in the cytoplasm. okay? Can't be synthesized within the rough endoplasmic reticulum, because if you look, once it goes in here, this, once it's enclosed within this membrane structure, this membrane structure is like two different locations, okay? So if you watched the last video, I did talk about that certain organelles are formed by the infolding of membranes. Well, those proteins, if they're within these places, then they kind of tend to stay within organelles, or they kind of tend to stay within organelles. All right, any protein synthesized in the cytoplasm generally tends to stay in the cytoplasm. Okay, so now let's look at what happens to the mRNA once it's bound the ribosome. So if the mRNA is bound to a cytosolic ribosome, then the ribosome will read the mRNA codon by codon, and according to the codons that it reads, specific tRNAs will bring specific amino acids into the ribosome to be joined in the growing polypeptide chain, and the amino acids, the sequence of amino acids that it puts together are dependent on the codons in the mRNA, and the codons the sequence of the codons is dependent on the sequence of the triplets that were present in the gene. Okay, so anyway, the ribosome then begins to synthesize a polypeptide, and we'll draw our polypeptide in blue. So any protein will draw in blue. So our polypeptide begins to get synthesized there, and then it starts to fold up in, and then the ribosome will move along the mRNA until it gets to the stop codon, and the polypeptide then detaches and then starts to fold up into its secondary structure, and then its tertiary structure, and then ultimately adopts its final tertiary structure, possibly quaternary structure, and can then function. Okay, so if the mRNA binds a ribosome in the cytoplasm, it will get translated by that ribosome into a polypeptide. The polypeptide then starts to fold up in the cytoplasm into its tertiary or possibly quaternary structure and adopts its final shape. Now that protein is now fully functional and will go around colliding with things in the cytoplasm, potentially sometimes colliding with its actual substrate, and will carry out its catalytic role in the cytoplasm. Okay, so that's the end of the story for that cytosolic protein. But what happens to the protein that binds to the ribosome on the rough endoplasmic reticulum? Well, that ribosome will synthesize the protein, but the protein will end up within the lumen of the rough endoplasmic reticulum. So that ribosome will be reading the mRNA on the outer surface, but the polypeptide will be being synthesized within the lumen of the rough endoplasmic reticulum. So that some proteins that exist as soluble proteins within organelles, they get synthesized completely within the lumen. But there are some proteins who are destined to be within the membrane of organelles. So either the membrane of organelles or the membrane of the cell surface membrane. In that case, something special happens, that those, let's say we have a different protein, let's say we have a different mRNA, and that mRNA was binding to this ribosome here. Now the protein that's supposed to be in a membrane, again, it has specific sequences within the mRNA that tell the ribosome to synthesize it in a slightly different way, or that end up being synthesized in a different way. And so if a protein is to be in the membrane, it gets synthesized so that parts of it are above the membrane and parts of it are looping through the membrane up and down like that. So part of it go through the membrane. I guess those are hydrophobic R groups there that will be okay to stay within that hydrophobic region of the membrane, and these parts are hydrophilic. So within the polypeptide, within the amino acid sequence, you've probably got a long stretch of amino acids that are hydrophilic, followed by hydrophobic amino acids, followed by hydrophilic, followed by hydrophobic. And that allows these proteins to go from one side of the membrane through the membrane to the other side as well. And thus we have our membrane spanning proteins that do various roles or jobs, such as transport proteins, such as receptor proteins, signaling proteins, things like that. Okay, so that's the different kind of proteins that you could have. Now let's follow what happens to them and how they end up in their roles. Because this protein right here is destined to function in the lysosome, right? Soluble within the lysosome, that's its end destination. How does it get there? And what happens to it on its journey? This membrane protein is eventually going to end up on the cell surface membrane, doing a very important job, being a receptor of some kind, allowing communication with other cells, responding to hormones, things like that. Okay, so how does it get there, and what happens on that journey? Then what happens next is that within the endoplasmic reticulum, carbohydrate modification begins. No. What first happens, and the most important thing that happens, is that proteins within the endoplasmic reticulum help the polypeptides to adopt their tertiary and quaternary structure. So these proteins, I'm going to call them chaperone proteins and just give them a blue C there. Remember, any protein is blue, so in the diagram. So these chaperone proteins, so some of the chaperones are attached to the membrane, so they probably help membrane proteins to help them fold into the final shape that they're supposed to be in. Some of the chaperone proteins will exist in the cytoplasm. Some of the membrane associated chaperones might help, not cytoplasm, sorry, the lumen of the endoplasmic reticulum. So they'll be helping these proteins to adopt their tertiary structure. So by the time that these proteins, now they'll move through the endoplasmic reticulum, the next key thing that happens to them is that any carbohydrate modifications begin here, right? So this protein, for example, is going to end up with a carbohydrate modification, as many proteins do that have a face or have a side that faces the outside of the cell. And that process of carbohydrate modification begins in the endoplasmic reticulum. Okay, so if it's here, for example, how did we draw it? It's got it into out 1, 2, 3, 4, 5, like that. By the time it gets here, there'll be enzymes in the endoplasmic reticulum lumen that will start to add then carbohydrate modifications. Not all of these modifications stay there. Some of them, as they go through the Golgi, they get removed. But most proteins that go through the rough endoplasmic reticulum do get carbohydrate chains added to them. And some of the purpose of that is that, well, it's important for its end role, but some of that purpose is that these chains help the polypeptide to fold correctly. Okay, so what happens then is that these proteins make their way to the ends of the organelles. So 1, 2, 3, 4, 5. Okay, so it's got to the end there. It has a carbohydrate modification. Yeah, and now it's time to move on to the next stage of its processing that's going to happen in the Golgi apparatus. Okay, so I've just removed part of the diagram here, just so that it makes the next part a little bit more clear. But the next thing that's going to happen is that these proteins now move on to the Golgi apparatus, where further modifications will happen, and these proteins will be packaged into vesicles that will take them to their end destinations. I had a bit of a recording incident, so things might look a little bit strange. So all the modifications that begin in the rough endoplasmic reticulum are very general. So it doesn't really matter what protein it is, the same modifications are slapped onto them. However, In the Golgi apparatus, the modifications continue, but these modifications are extremely specific to the protein and its eventual function. And we'll talk about how that is achieved a little bit. Okay, but let's talk about how the proteins get to the Golgi apparatus. Essentially, the protein, the ends of the endoplasmic reticulum, right there, they pinch off. So proteins attach to the outside of these membrane areas. They attach and cause the membrane to bend, and that requires energy. And so that's another reason why the cell is so dependent on having ATP around at high levels all the time. So energy is used to bend the membrane, deform it, and pinch it off its compartment. And so it's done here. So it contains the soluble lysosomal enzyme that's going to end up in this compartment here. Whereas here, this membrane protein is in the membrane of that vesicle. Okay. Now, while we're here, well, let's not forget, as I did in take 1, that we also have this secreted protein here. So this protein is doing its job outside of the cell. And how it's going to get there is following a similar route. So what we'll do is we'll just include it here. So, for example, it might be in one of these vesicles. So it'll be synthesized, and it might end up in that vesicle also. Okay, just hypothetically. So it will be contained within that vesicle. All right, and we'll follow its journey as it goes through here. Okay, likely secreted proteins also have some modifications on them, some carbohydrate chain added. Okay, so it could be a hormone, peptide hormone, it could be an extracellular enzyme, it could be an extracellular structural protein such as collagen, it could be an antibody. Okay, but there it is in that vesicle. And now what happens with these vesicles is that they arrive, they are transported. So these are transport vesicles. Okay, and they arrive at the Golgi apparatus. And when they get here, again, their membrane goes through the opposite process of fusing. Again, energy is needed. You've got proteins that are pushing these membranes together or pulling them, however you want to look at it. But they're bringing these phospholipid bilayers together until they are one with each other. And so not only is that protein delivered here, and this protein will be ending up, so if this happens here, if this vesicle fuses with this membrane, the soluble protein will end up in here, okay, with its little bit of modification, okay, but that membrane protein will end up in the membrane, 1, 2, 3, 4, 5, in the membrane of the Golgi apparatus. And so any modifications that would be done by enzymes in this, be done here. So the modification faces inward here. What happens next is the modifications then begin. Now, as I said to you, the Golgi apparatus carries out specific modifications, very tailored to the nature of the protein. Now, on what basis does it decide to modify one protein in one way and another protein in another way? And again, it comes back to the sequences. This is why we say that the nucleus controls everything, is because everything comes back to the genomic information in the DNA. So here, these proteins have very specific amino acid sequences. So based on these specific amino acids, you've got enzymes here, and we'll call these enzymes modification enzymes. So we'll call them M. And the special thing about the Golgi apparatus is each of these different cisternae, okay, that's the cis phase, that's the trans phase, and these are the different cisternae. Each of these cisternae has a different subset of modification enzymes. So we'll just denote them M2, M3, M4, and M1, just kind of symbolizing the idea that we've got different enzymes floating around in this lumen compared to this lumen, compared to that lumen. So different modifications will happen as the proteins move through these different stacks. Okay, so there we have that. And so how is it that these modifications can be specialized? Well, each of these proteins is an enzyme, and enzymes recognize very specifically their substrate. In this case, these enzymes will recognize the amino acid sequences as their substrate. So if this enzyme doesn't recognize any of the amino acid sequences on the surface of this protein, it cannot act therefore as a substrate to this enzyme, and therefore this enzyme won't add any modification to that protein. Whereas if this enzyme can recognize some of the amino acid sequences on these proteins, it will go ahead and act on those proteins as a substrate and modify their modifications. Okay, so that's how these enzymes can be specifically doing different things to different proteins, or different things to different proteins can be done within the Golgi apparatus, because you've got these enzymes that recognize them as something to be done something with or not, based on their amino acid sequence. Okay, so remember that ER, or rough endoplasmic reticulum, very generalized modification begins, but Golgi apparatus specific modifications tailored to the type of protein that's coming through. Okay, and the purpose of there being all these different compartments is that there's different enzymes housed in those compartments. So the way they move from one cisternae to another is again by the same process, but it doesn't happen through here, it kind of happens from the ends here. So this protein will arrive at the end. It's clearly not been recognized by any of the enzymes here, so it goes through unmodified. Okay, pinch off in a vesicle that contains it. Okay, and then fuse and thus be delivered to the next cisternae. Okay, so there we have that. Right, and so the process continues until it gets to the end, and if no further modifications are needed, it is then packaged up into, it then gets packaged up into another transport vesicle, and we call this a transport vesicle still because it's being transported still within the cell from one compartment to another. So we're going to call that a transport vesicle. So the protein is there. Okay, and again, proteins are very actively deforming the membrane, allowing it to pinch off. Okay, and we get then a transport vesicle, which will then move towards the lysosome. And when it moves to the lysosome, again, other proteins will be involved in fusing these membranes together so that the content of the vesicle can then enter and then work in the lysosome, performing its function. So in the lysosome, you've got hydrolytic enzymes. So this is likely some kind of protease, lipase, carbohydrates, nuclease, one of those enzymes. So anything that needs to be degraded then. So if I've got a protein that's, a little bit mashed up, there'll be vesicles that will be being also transported to the lysosome, because we have maybe a protein that is non-functional, and its amino acids are just being wasted because it's not doing its job anymore. So these vesicles will then be transporting cargo to the lysosome because it needs to be degraded. So now we've got the enzyme that's going to do the degrading. Now its substrate has been delivered, and put those two together and you get enzymes. Not enzymes, you get amino acids. So that protein's been broken down into amino acids, which then the cell can reuse. Okay, we also want to note that not only has the cargo been delivered, but the lysosome has now got all this extra membrane that's part of its structure now. So this is probably also the delivery of vesicles from one place to another is also a way in which these organelles grow themselves. Yeah, so not only is stuff being transported to them, but they now have all this extra phospholipid bilayer that's allowed them to grow. And that's probably the way in which they grow, to the point which at the time of cell division they're big enough to be distributed to different cells. Actually, we've discussed how cytosolic proteins end up working in the cytosol or cytoplasm, how lysosomal or compartmental proteins end up at their respective compartments. What we haven't done is looked at the cell surface membrane and secreted protein. So we'll look at that now. So as these proteins go through, so this protein will be modified, it will pinch off in a vesicle, it will end up at the next stage. Now 1, 2, 3, 4, 5 proteins that are glycosylated, they go through a very complex procedure in the Golgi apparatus. So their original general modifications might be removed, trimmed down, and new and improved, better modifications specific to those protein functions are now added. Okay, and then once that's done, it moves through again, pinching off, fusing, pinching off, fusing, and then finally pinching off again into this time secretory vesicle. Okay, secretory vesicle. So these ones essentially, look, they're performing a very, very similar function. It's just because these vesicles are headed towards the cell surface membrane, and they're going to be called secretory vesicles, and usually their cargo is usually quite electron dense. So in an electron microscope, you'll see that they're very dense with the stuff that's been packaged into that vesicle. Okay, right. So now our membrane protein, 1, 2, 3, 4, 5, our membrane protein with now its new and improved, perfectly suited to its function, carbohydrate modification, is now going to end up, as this pinches off, so as this pinches off, this is now contained in the membrane of this vesicle. 1, 2, 3, 4, 5. Carbohydrate modification. Okay, so we have that there. Also, this secreted protein is also going to be in there. This protein is going to be secreted. Okay, and then this secretory vesicle is going to make its way to the cell surface membrane. So just so that we understand how this happens, we have our 1, 2, 3, 4, 5. We have our membrane protein that's destined now for the cell surface membrane with its now perfectly finalized carbohydrate modification. And in a similar, I'm not saying that these are always together, but in a parallel situation, we might also have our secretory or secreted protein right there with a little bit of modification of its own there. And now these two membranes are going to fuse. Okay, and so we have now our membrane protein that began its journey. I don't know if you can see everything still. Right, and we have our membrane protein that began its journey over there, synthesized within the membrane of the rough endoplasmic reticulum, and now it finds itself in the cell surface membrane. And if we were to just pull this membrane a little bit, let's just see what would happen. So if we did just pull that membrane and helped the new arrival settle into its new destination, it would look like this. Okay, so 1, 2, 3, 4, 5. And our secreted protein would now be floating around in the tissue fluid. Okay, so whatever fluid was in there now is there. Okay, and so maybe the cell loses a little bit of volume in doing that, but remember, there's also a lot of endocytosis happening, bringing fluid into the cell, and just as membrane is being added to the cell by this process, endocytosis helps to bring membrane back into the cell similarly. Right, okay, so let's not forget our carbohydrate modification. And there we have it, okay? So that's our 4 proteins. If you remember, at the beginning of our story, we had a cytosolic protein produced, maybe an enzyme that's involved in glycolysis. So that had a very short, straightforward journey, synthesized in the cytoplasm, and there it works. For proteins that have a job to do in a specific compartment, they go through the rough endoplasmic reticulum, through that pathway of transport vesicles, okay, being modified along the way. And again, membrane proteins exist within the membranes of these compartments throughout their life, and they have usually a lot of extensive modification done through that process, as do secreted proteins. And there we have that. So essentially that was a look at the functions of all the organelles. Oh no, one more thing, guys, as long as we're still rolling. One more thing, and this is the question, and I think it's not an obvious question, but where does all this lipid come from? Okay, so in order for a cell to grow, clearly all this membrane needs to come from somewhere, and this is where the smooth endoplasmic reticulum comes in, because we talked a lot about membranes, proteins being synthesized in the membrane of the rough endoplasmic reticulum, bits of the membrane then pinching off, forming vesicles, taking proteins to different compartments, and the membrane of the different compartments, like the lysosomal membrane, probably originated mostly from the Golgi. And the membrane in the Golgi, a lot of it probably came from vesicles arriving from the rough endoplasmic reticulum. So where did the rough endoplasmic reticulum membranes and then nuclear membrane, where did all that stuff get produced? And there we have the humble smooth endoplasmic reticulum, which is a structure very, very closely related to the rough endoplasmic reticulum, except for it has a different structure and a different function. It's got interconnected membranes. We'll come back and discuss the structure, but let's talk about its function. Its function is, guys, its function is to synthesize the membrane that maintains all of these organelles, that allows things to be transported between them and the production effectively of and maintenance of the cell membrane. So all these phospholipid bilayers were made here, okay? Because what happens, the function of the smooth endoplasmic reticulum is that special proteins in the cytoplasm deliver fatty acids. Yeah, let's keep that in black because all the lipid related stuff will. Okay, so proteins deliver fatty acids to the lumen of the smooth endoplasmic reticulum, and also glycerol. And what happens is that enzymes within the smooth endoplasmic reticulum join up the glycerol and the fatty acids to form lipids, to synthesize the phospholipids and all the other specialized lipids that go on to form the membranes, including cholesterol. Okay, so all of that is formed within the smooth endoplasmic reticulum, and so this smooth endoplasmic reticulum, as well as forming hormones and things like that, and also in specialized cases being the container for the calcium ions. But the main role of this organelle is to synthesize the phospholipid bilayer. So all the smooth endoplasmic reticulum is essentially, it's a membrane factory. So all the membrane that gets produced here allows the smooth endoplasmic reticulum to grow. But then because the smooth endoplasmic reticulum is kind of connected and joined and continuous with the rough endoplasmic reticulum, as it makes more membrane then gets kind of distributed to the rough endoplasmic reticulum. And as the rough endoplasmic reticulum produces these vesicles that pinch off, it delivers all that membrane to the Golgi apparatus and allows the Golgi to grow. And as the Golgi grows, the membrane pinches off in these vesicles, so the membrane that was produced here then is making its way to these other organelles and allowing the cell membrane to also get new membrane. So, smooth endoplasmic reticulum, we kind of take for granted, oh yeah, it synthesizes lipids, but let's not forget that essentially that is what causes the formation of all these other membrane compartments. So we've discussed the kind of main business, as it were, but while we're here, and since we've already invested so much time, why not go the whole way and do this thing properly and consider how all the organelles of the cell might be contributing to this process. All this vesicular movement, all this vesicle transport is not possible because these vesicles, if they were just floating around in the cytoplasm, they would randomly end up anywhere, and they will eventually get to where they're supposed to go because they have proteins on their surface that will only latch on to the compartment that they need to be. So given enough time, the vesicle would eventually collide with wherever it's supposed to go, but that might take too long. So vesicular transport actually occurs, and I'll use a different color. Actually, the color that's most associated with this is green. So where's my green pen? Vesicular transport occurs along the part of the cytoskeleton. So generally speaking, the cytoskeleton is what allows a framework, some kind of tram lines along which these vesicles can move more efficiently to the place where they're supposed to go, and in a more directed manner rather than just relying on random collision. Okay, so we've got these microtubules. So part of that cytoskeleton is the microtubule network, and it's along the microtubules that the vesicles are moving to take them to where they need or where they are supposed to go. Okay. All right, so we have that. So it's important to realize that even the cytoskeleton is involved in the story that we were talking about, first of all. Okay. The other thing I wanted to quickly mention was energy. We discussed that any biosynthetic or all these membrane pinching offs and all the membrane or the vesicles fusing with membranes, allowing transport of contents from one compartment to another, all of that required energy. So there has to be some mechanism by which ATP, the energy currency that's being used by all the active processes in the cell, there must be a way for ATP to be generated, and that's where the organelles that we've largely ignored up to now, that's where they come in to it. So eventually we will talk about the chloroplast and the mitochondria in a future episode, but suffices to say that without organelles carrying out the functions of chloroplasts, so let's say that's our mitochondria there, stalked particles. So it is there, it's floating around, and thanks to those organelles we have ATP, and thanks to ATP we then can carry out the transport, the membrane fusions, the membrane pinching off, I don't remember what that's called, but all those processes require energy, and thanks to mitochondria and chloroplasts we have that energy. Okay. What else? Also glucose, in order that we can build these molecules, in order that we can have fatty acids, in order that we can have glucose molecules, monosaccharides in which to build nucleotides, in which to synthesize amino acids, we need glucose. And so it helps that we have chloroplasts perhaps in plant cells that can build those molecules up. Okay, so our undertaking of the discussion of the cellular organelles isn't really complete just talking about the function. The function sets the scene. Now is the best time for us to look at the structure of the organelles, because we know what they are doing. And because we know what they are doing, we are in a better position to talk about how their structure relates to their function, because essentially the structure is the adaptation of the organelle to perform its function. Okay, so just watch out for that. Examiners might directly ask you for the structure, or they might ask you for the adaptations of the organelle that allows it to perform the function. I think essentially in most cases they're asking for the same thing. Right, so the nucleus houses the genomic information in the form of DNA, which when we look at DNA, we'll look at how the DNA is organized into chromosomes. We'll look at its specific structure. Okay, but the DNA is there in the nucleus, in chromosomes. Remember that there's homologous pairs of chromosomes in this nucleus. But the point is that, as we discussed before, all the compartments of the cell have their own proteins that give them their characteristic structure and functions, but the information to produce those proteins is all in the chromosomes of the DNA right here, or all in the DNA of the chromosomes. So we have all the genetic information stored here. Now, actively expressed genes, it takes a lot of it takes a lot of proteins in order to carry out transcription. So wherever there's lots of transcription happening, you're going to get lots of RNA, and you're going to need lots of proteins. For example, you have RNA polymerase, you have transcription factor proteins. All of those proteins will be needed to carry out or to copy the DNA into RNA. Okay, so wherever that's happening, under an electron microscope, those regions are going to appear a bit darker because there's so much more protein content in that area. Okay, now there's a particular part of that genetic mixture that has a lot of actively transcribed genetic information, and the area of the nucleus that has the genes needed to produce the ribosomes, that is the most actively or the most active area in terms of gene expression, and so it has the most proteins all huddled up together expressing the genes needed to make the ribosomal RNA, the ribosomal protein component. So they're making the mRNA there, but not only that, the protein part of the ribosome comes back into the nucleus. So the protein is made out here, just like all the other proteins. But then the protein part of the ribosome comes back in and gets combined with the ribosomal RNA here, and then the whole ribosome gets exported back out again. So there's a whole load of ribosomal RNA, ribosomal proteins, the proteins needed for the transcription of all the ribosomal genes, all in this area. And this area therefore stains or appears very, very dark under the in an electron microscope image, and is therefore known as the nucleolus. Okay, nucleolus. So we have the nucleolus here, so we have lots of genetic information. The more active areas need more proteins and they appear darker, and the darkest of all the areas is the nucleolus, because that's where the ribosomes are being synthesized. Okay, so we have that. So moving outwards then from there, we have the inner membrane of the nucleus. Okay, so it's a double membraned structure. It has an inner membrane and an outer membrane. The inner membrane, its specific structure is that it has specific proteins that are needed for attachment to the chromosomes. Now that's quite important, because during the cell division process, you need the nucleus to detach away from the chromosomes, and then once the cell division has happened, you need the nucleus to reform around the chromosomes, and it's these proteins on the inner membrane of the nucleus that allow that process to be controlled. So when it's time to detach, those proteins are affected, changed, modified so that they detach from the chromosomes. And when it's time to reform the nucleus, those proteins are modified again so that they can attach back to the chromosomes and the nucleus then reforms. So there's that on the inner membrane of the nucleus. As well as that, there's a form of cytoskeleton inside the nucleus called the nuclear lamina that gives shape, support to that nucleus structure. Okay, and now we move outwards and we have the outer nuclear membrane. Now the outer membrane of the nucleus is joined to the membrane, or is connected or continuous with the membrane of the endoplasmic reticulum. So you're going to have a few ribosomes also on the outer nuclear membrane. Okay, and then between them we have these nuclear pores. Now nuclear pores are not just membrane pores or membranous pores. They are composed of many proteins, hundreds of proteins that are organized to form this highly regulated protein structure that's got a very specific role of controlling which molecules can move in and out of the nucleus, because that genomic information is very important to maintain. Okay, so only things that are needed to express the genes can go in, and any damaging proteins, such as potentially digestive enzymes, cannot be allowed to freely go in to the nucleus because it will result in degradation of the genetic information. So that movement in and out has to be controlled. Okay, however, small things like hormones, small molecules, nucleotides, for example, they can easily go in and out via passive diffusion. However, it's molecules, large molecules such as ribosomes, such as DNA polymerase. So DNA polymerase is an enzyme, so it's going to be synthesized out here, but in order to replicate the DNA, it needs to move into the nucleus. RNA polymerase needs to move in to the nucleus. Histone proteins made out here need to go into the nucleus. So all those proteins need to go in, and they do so via a very controlled mechanism, via the nuclear pore complex of proteins. Okay, so it's not just a hole, it is a machine, that nuclear pore, okay, allowing the controlled movement in and out of the nucleus. Okay, now moving on outwards, then we have our endoplasmic reticulum. Now the rough endoplasmic reticulum is always shown as a series of interconnected flattened membrane sacs, okay, covered with ribosomes. Clearly, because it's got an important role in protein synthesis, those sacs are flattened, covered with ribosomes, so it can synthesize proteins, flattened to increase surface area. Okay, as that's a continuing theme throughout biology, so make sure that you're aware of that. The smooth endoplasmic reticulum is not involved in protein synthesis, so it does not have ribosomes. However, while they are still interconnected and they're continuous with the rough endoplasmic reticulum, the smooth endoplasmic reticulum is usually interconnected tubules, or described as interconnected tubules rather than interconnected flattened sacs. Okay, so just be aware of those descriptive differences. Okay, vesicles of all kinds, they're pretty straightforward. They are bound by a single membrane, a membrane of phospholipid bilayer, so be aware of that. So I've drawn it as one line here, but remember all membranes phospholipid bilayer, and their contents are the same as the organelle from which they came from. So the fluid here, if this organelle pinched off from there, or if this vesicle pinched off from here, its fluidic contents would be very similar to that. Okay, so we have single membrane bound vesicles, and then we have the Golgi apparatus. So structurally speaking, it is a series of so we're just gonna throw out the word interconnected because they are not interconnected. A series of flattened membrane sacs, each called cisternae. There's a cis face facing the nucleus, there's the trans face facing the cell surface membrane. And I think that's really all there is to it. Structurally speaking, remember we discussed that what allows the modifications to take place is the fact that there's specialized enzymes within the lumen of the cisternae, and different modifications are carried out in the different cisternae by having a different composition of enzymes in each of the cisternae. Okay, so that's essentially the Golgi apparatus. I can't really think of anything else relating to its structure there. Lysosomes, they are essentially, they're just glorified vesicles in a way, but structurally speaking, it's got a single membrane that allows its contents to be separated from the rest of the cell, which for the lysosome is really, really important because you don't want these hydrolytic enzymes to be spilling out into the rest of the cell because it's just going to break everything else down. Okay, it's going to be the focus of another lesson or video where we discuss the energy organelles, the chloroplast and the mitochondria. They deserve their own because remember they have a different evolutionary origin, so we can compartmentalize them or group them or classify them according to that. So here we have discussed the organelles of the cell involved in the synthesis of lipids and protein, and next time we will discuss the energy organelles or the endosymbiotic organelles. Okay, and they're mainly to do with the energy requirements of the cell. All right, guys, so that will be the next episode. Thank you for watching. All right, and I do appreciate that you've stayed to the end. You are the real biologists, guys. So well done for that. Well done for persisting. Right, keep going. Next time, remember chloroplasts and mitochondria. Any questions, guys, do tweet me. So any questions, I do want to make videos where there's some form of me answering your questions. So either if you have a question or anything about this, please do put it in the comment section and I will respond, either respond to you via messaging or I'll make a video if it is needed explaining the answer to your question. All right, guys, thank you for watching and I will sign out. Good luck, guys.